So, dear colleagues, today is the day of our seminar, and it's my great pleasure to introduce our speaker, Professor Torsten Steinmetzer, a leading figure in the field of pharmaceutical and medicinal chemistry. He currently holds the position of full professor of pharmaceutical chemistry at the Philipps University Marburg in Germany, a role he has held since 2007. His academic journey began with a degree of biochemistry, not in chemistry, biochemistry, from Martin Luther University Hall, followed by a doctorate in biochemistry. He later completed his habilitation in Friedrich Schiller University Jena, focusing on the development of synthetic thrombin inhibitors, an area that would become a cornerstone of his research career. With over 3 decades of experience spanning academia and industry, including leadership roles in 2 companies, Professor Steinmetzer has made a contribution to the design and development of protease inhibitors. His work has significantly advanced an understanding of serine proteases. He is an author of more than 100 peer-reviewed publications, many of which have shaped current approaches to drug design targeting proteolytic enzymes. His research is characterized by a deep integration of synthetic chemistry, enzymology, and structural biology. And okay, floor is yours.
Okay, thank you very much, Valentin, for this kind introduction. And at the beginning, I have incorporated 2 slides simply to show a view on Marburg. Marburg is a relatively small city. We have around 73,500 inhabitants, and it is located 1 hour in the north from Frankfurt by car. And you see 2 landmarks of Marburg. This is the Landgrave Castle on the top of the hill. And the second very famous landmark is the Elizabeth Church here on the right side of this figure. And our institute is located in the center of the city in this area. And I would like to speak today about the development of furin inhibitors. Originally, we started with peptidic derivatives, and later now in the present time, we are dealing with non-peptidic inhibitors of the serine protease furin. These are 2 additional photos from Marburg. This is the Lahn, this is the river where Marburg is located. And this again is a photo from the Elizabeth Church. The construction started in 1235, only 4 years after the death of the Saint Elizabeth. And it is the first Gothic, purely Gothic church in Germany. Yes. And this is a photo of the probably most famous person from Marburg. This is Emil von Behring. He was awarded in 1901 with the first Nobel Prize in medicine for the development of the passive immunization, yes, the serum therapy, where he treated diphtheria patients and tetanus patients. And in this red building, there is probably living the second famous guy from Marburg. It's the Marburg virus. Yes, this is a photo of the biosafety level 4 lab in Marburg. It's the only BSL-4 lab at the German university. And the Marburg virus was imported from Africa in 1967 by the Behringwerke. They imported some monkeys which were infected by this Marburg virus. And as a consequence, 30 people in the lab were infected and 7 of them died due to hemorrhagic complications. Yeah, it's similar. It's a filovirus like Ebola virus. And meanwhile, on the right side of this building, they built or started with the construction of a new BSL-4 lab, and it should be finished in 2 years in 2027, because in this year, the University Marburg celebrates the 500th anniversary of its foundation. It was founded by Landgraf Philipp in 1527. So this is the 500th university, and then we obtain a new BSL-4 lab. And since that time, Marburg has a relatively strong institute of virology. And I have a long collaboration with the colleagues from this institute because several viruses depend on proteases from the host for their replication mechanism. And as a medicinal chemistry, you need collaborations, you need people which are testing your compounds. And I have simply shown it in this slide. This should be an enveloped virus. You have a lipid envelope. And in this envelope, the virus carries some surface proteins. And very often, these surface proteins have to be activated by proteases. Yes? And many viruses don't have an own protease, and they use proteases from the host for activation of the surface proteins. And the cleavage of these surface proteins is a prerequisite that the virus membrane can fuse either with an endosomal membrane or can fuse directly with the surface of a cell. Yes? And as a consequence of this fusion of the viral membrane with the endosomal membrane, a fusion pore is opened and the content of the virus, also the nucleic acids or the proteins, can be released in the cytosol of the infected cell. And this is a prerequisite for replication. And there is one protease very often involved in the activation of many human pathogenic viruses, and this is the serine protease furin, which cleaves peptide sequences after multi-basic amino acids. Yes, this is the cleavage sequence. R is the one letter code for arginine, K for lysine. And many viruses are dependent from furin. For instance, the highly pathogenic bird flu viruses of the subtypes H1 and N1 and H7N1. And in the moment in Germany, we have a bird flu outbreak. And also certain coronaviruses depend on furin, for instance, the new SARS-CoV-2 coronavirus. Yes? Also, all flaviviruses like dengue virus, West Nile virus, Zika virus, tick-borne encephalitis virus, or yellow fever virus need an proteolytic cleavage of their surface protein M by furin for replication. Also, chikungunya virus and Ebola virus, Marburg virus, HIV, measles virus, mumps virus, they are all depend on furin. And moreover, furin can also activate many bacterial so-called AB toxins like Shiga toxin, diphtheria toxin, Pseudomonas exotoxin, anthrax toxin, and therefore inhibitors for furin can be used also to neutralize these toxins. And in addition, furin supports also tumorigenesis, at least for some tumors. And it's clear, based on all these functions of furin, an inhibitor could be useful as anti-infective agent, and at least also as a broad spectrum anti-infective agent, because you do not only target one virus, you would target all these viruses with an efficient furin inhibitor. Furin belongs to a special family of serine proteases, which is called the human proprotein convertases. These are calcium-dependent proteases, which contain a subtilisin-like protease domain. And the normal function, the physiological function of the PCs is to activate many proforms of proteins from hormones, from receptors, from enzymes and other proteins. And the family contains 7 furin-like PCs, which have a similar substrate specificity like furin. It means they cleave after basic sequences, preferably after an arginine residue. Some of them are membrane-bound. Other of these PCs are secreted into the bloodstream, and some PCs exist in secretory vesicles like PC1 and PC2. And then we have an additional member which is named S1P, the site 1 protease. Another name is Ki1. This also cleaves after non-basic residues, and it's involved in the cholesterol metabolism. And the last member of this PC family is PCSK9, and it's interesting. It has a sequence of a protease, but it's not proteolytically active because it contains a pro-domain. This is cleaved in an autocatalytic manner, but it's not released. And therefore, this protease is blocked by its own pro-domain, so it's not an active protease. But despite this, it's an interesting target for drug design because PCSK9 binds to the low-density lipoprotein receptor. And if this complex is formed, the low-density lipoprotein receptor is degraded and removed from the cell surface. And as a consequence, if you have less of this receptor, you can fish out a reduced amount of low-density lipoproteins, which are responsible for arteriosclerosis from the bloodstream. And therefore, inhibitors of this PCSK9, which block the binding to the LDL receptor, can be used for the treatment of hypercholesterolemia. Yes? And there are 2 antibodies on the market, and inclisiran is an siRNA against PCSK9. Okay, we started many years ago with the development of these furin inhibitors, and I want to explain the whole way, so simply to come to our newer compounds at the end. So the first inhibitors, which were relatively potent, were synthesized by one of my first PhD students here in Marburg. It was Gero Becker, and I gave him such an benzamidine residue, which I used in previous time very often for the synthesis of inhibitors of trypsin-like serine proteases, which is simply a different protease class. The clotting proteases belong to the trypsin-like serine proteases, thrombin factor 10A. And it was very surprising that one of these first compounds was already a very strong furin inhibitor with a Ki value of 0.8 nanomolar. And the inhibitor consists of 5 groups, which are numbered here or named as P1, P2, P3, P4, and this is the P5 residue. And it's like a building block system, like a Lego system. You can replace individual residues, and then you can identify the best residues for each position. And at the end, you combine the best residues, and then you can improve the inhibitor. And as I mentioned before, it was a nice surprise that this compound was already so potent. The synthesis is relatively straightforward, no problem. And we had also a relatively high selectivity for the family of proprotein convertases, for these proteases, because the compound had a relatively poor potency against trypsin-like serine proteases. But of course, it's a peptide compound, so you have probably a relatively poor cell permeability, and because it contains several amino acids, it might be possible that it is degraded by proteases. Then we further modified this compound. We simply substituted this P5 residue with an additional basic residue. We incorporated such a guanidino-methyl group, and we could significantly improve the potency. We incorporated this in this C3 position or C4 position, and we were coming to compounds with Ki values in the low picomolar range. And for this inhibitor, we also obtained a crystal structure from our cooperation partner Sven Dahms. At that time, he was still in Jena in the group of Manuel Tan. And here you see the furin, which is colored by its electrostatic potential. In red, you see an acidic region, as a negatively charged region, and in blue, these are basic region. And the active site cleft is strongly acidic because around the bound inhibitor, you see 13 acidic amino acid residues, also 13 glutamic acid or aspartic acid residues. And that's why it's not surprising that such multi-basic inhibitors are strong inhibitors of furin. And then we also had a look on the so-called P3 position. And I was very surprised at the beginning when Gero Becker told me that the valine and isoleucine derivatives are more than 10-fold more potent than the leucine inhibitor. Yes, so we have a Ki value of 12 nanomolar. Here we are below 1 nanomolar. And we didn't understand it because the side chain of this P3 residue goes out from the furin surface and is directed into the solvent. So it was hard to explain why this leucine containing inhibitor is less potent than the analogs with valine or isoleucine. And what is also important, this P3 residue makes an antiparallel beta sheet interaction with a furin residue glycine 255. Yeah, this is very important, this contact. And then with an next PhD student, Cornelia Hardes, we stepwise modified the P3 side chain simply to understand what is the best residue in this position. So we started with glycine. All these proteinogenic amino acids are colored in blue. The non-proteinogenic amino acids are colored in black. And below I have always written the Ki value in the unit nanomolar. If we attach simply a methyl group, we are coming to alanine. We improve the potency by a factor 3. If we add an additional methyl group, we are coming to the aminobutyric acid derivative. It's further improved the potency. And valine is even more potent. Here the value is a little bit lower than in the slide before because Cornelia made all the measurements new, and in previous measurements we determined 0.8 nanomolar. So there's no much difference. And then we elongated here the side chain of the aminobutyric acid. We are coming to a norvaline. It has similar activity. The isoleucine I have shown before, it's also very potent like the valine. But the most important improvement was when we added an additional methyl group to the side chain of valine, and we are coming to this tertiary leucine. Another name of this amino acid then is tertiarbutylglycine. Yes? And also very potent was a derivative where we incorporated penicillamine, and these potent amino acids are all beta-branched amino acids. Yeah? Leucine, in contrast, is a gamma-branched amino acid. And this is the important thing. We need beta-branched amino acids to improve the potency of these compounds. And when we found this, of course, you start to read publications, and you see several reports where the people incorporated tertiary leucine as an substrate analog peptidic structures. And it has a space-filling side chain, and as a consequence of that, it stabilizes beta sheet backbone conformations. And it's important to know that all substrates and all substrate analog protease inhibitors bind in form of an antiparallel beta sheet to their target. Yes, and if you stabilize the beta sheet conformation, then you get an improved potency. And it improves also the stability of peptides. And therefore, it's not surprising that tertiary leucine is used in several marketed protease inhibitors. For instance, in the inhibitors of the hepatitis C virus protease, there we have the compounds boceprevir, telaprevir, glecaprevir. Atazanavir is an inhibitor of the HIV protease, approved. It even contains 2 tertiary leucine residues. And it's also a residue in nirmatrelvir. This is the new inhibitor of the SARS-CoV-2 main protease developed by Pfizer a few years ago. Yes, and there we have also as a P3 residue and tertiary leucine. So it's an interesting amino acid. And the Chinese people have also developed a very similar analog to nirmatrelvir. It's called simnotrelvir. This also contains this tertiary leucine residue. Then, of course, we combined the tertiary leucine with the basic substituents on the P5 residue. We incorporated not only the guanidinomethyl group, but also the aminomethyl group. And all these inhibitors possess a low picomolar potency against furin. And these are some crystal structures of this inhibitor 1148. These are the interactions in the so-called S1 pocket. This is this binding pocket on the enzyme, which is occupied by this benzamidine, which is the P1 residue. Yes? And we see a salt bridge from this amidino function to an aspartic residue, and then additional polar contacts to carbonyl groups from proline and an alanine residue. And below this S1 pocket, there is one of these important calcium ions, yeah, which stabilizes simply the protease conformation. And in the S2 pocket, there we have an arginine in our inhibitor. We see also several polar contacts. And here it's possible to replace also the arginine by a lysine residue. The activity drops a little bit by a factor of 2 to 3, but not very much. P3 position, we see this anti-parallel beta sheet interaction with this glycine residue from furin. And in P4 position, we have also an arginine. This is also involved in several salt bridges to glutamic and aspartic acid residues. And this is very important, this arginine. If we replace it here with a lysine, which has only an amino group, we lose approximately a potency by a factor of 200. Yes? So this is very important. And these are the interactions of the P5 substitutions. So we have a huge number of salt bridges and polar contacts between the inhibitor and furin, which explains the unusual high potency in the low picomolar range. And at that time, we were very happy with this compound. We could say we had the most potent furin inhibitor developed on the world. But the problem was when we, for the first time, obtained some results from a toxicity study in mice. And the mice tolerated only a relatively low dose of the inhibitor of 2.5 milligram per kilogram when it was applied intraperitoneally. Yes? And at the next higher dose of 5 milligram per kilogram, all 4 mice which were tested died. So this was very strong disappointment at that time. And we sent also some other compounds for toxicity testing. And we found, for instance, when we replaced the P2 arginine simply by a lysine, so then we have only an amino group here instead of a guanidino group. The potency is not very strongly reduced, but at least in this case, the mice accepted a dose of 5 milligram per kilogram. Yes? And also when we removed the amidino function, yes, we completely lost potency. So the Ki value was increased to 1 micromolar. So we lost nearly at least 100,000 or more on potency. And also this compound was only tolerated by 10 milligram. A little bit higher could be dosed, but it was also toxic at 50 milligram. And based on all these data, at least we hoped that the toxicity was not directly related to the inhibition of furin, because otherwise the project should be stopped. Yes, if the toxicity simply comes from the inhibition of furin, and we hoped that we address with these compounds an unknown off-target. Yes, but at that time we had no idea about which off-target it could be. At least the toxicity correlated more simply with the number of very strongly basic groups, guanidino groups, amidino groups, than with the inhibitory potency against furin. Therefore, we simply tried to get rid of this, or get rid at least of some of these very strongly basic groups. And one possibility was the replacement of arginine with this unusual amino acid canavanine, which contains an oxyguanidine group in the side chain. The electron withdrawing properties of oxygen lead to a reduction of the pKa value from arginine, where the pKa value is 13 around, to 7.04 in case of canavanine. So we have a very significant reduction of the pKa value. So it means at physiological pH value, this side chain is only partially protonated. And indeed, this helped to reduce the toxicity of this compound. And in that case, a dose of 15 milligram per kilogram was tolerated after intraperitoneal treatment in mice. Yes? At that time, no higher dose was tested because all these toxicity tests were done by a company in Taiwan, and the protocol was fixed before the experiment started, and we had selected at that time 2.5 milligram, 5, 10, and 50 milligram, so 4 different dosages, and so we could not go higher at this moment. We obtained also a crystal structure for this inhibitor, and surprisingly, always when we incorporate an canavanine SP4 residue, then we see 2 different conformations of the P5 group. Yes? This conformation, which is labeled here with A, this is the normal conformation, which we have seen with the inhibitors when we had simply an arginine in this P4 position. And now we have approximately to 50% a second conformation, but here there is no polar contact of this guanidino group with the furin surface. But it was, we have no real explanation why this is the case, but we have seen it with several analogs, not only with this one inhibitor. And this compound was synthesized by my PhD student, Thuy van Lam Van, which is the lady here on the right side of the photo. And this photo was done at the beginning of March 2020, and we are very lucky. We made this trip to the Kleinwalsertal only a few days before the lockdown in Germany due to the coronavirus infection started. Yes, we were coming back to Marburg on March 13, and the lockdown started on Sunday, and then all lifts were closed, and so we were really lucky at that time. Okay, of course, we tried also to modify the P1 residue because the benzamidine is also a very strongly basic group, and we synthesized many compounds, but nearly in all cases, we significantly lost potency of the inhibitors. But there was one interesting starting point, which was found by our collaboration partner, Sven Dams, which is determining all these crystal structures. He was at that time a postdoc for 6 months in our institute in the group from Gerhard Kleber. And he screened a library of small compounds. He performed a crystallographic fragment screen, and he found this amino isoindole as a fragment which binds exactly at the same position like the benzamidine in case of our inhibitors. And if you check the literature about amino isoindole, you find that it can exist in 2 tautomeric forms, nearly so at a 1-to-1 ratio. We have this isoindole amine, and the other is this isoindolin imine form. And we calculated also the pKa values. They are a little bit reduced compared to a benzamidine. In benzamidine, you find in literature values around 11 as a pKa value. So it's a little bit less basic at least this form. Also it was calculated with Marvin sketch with this pKa calculation possibility. And for the incorporation into inhibitors, we needed a modified amino isoindole, which contains here on this carbon, which is colored in green, an amino methyl group. And this simply shows you how we synthesized this compound. We started with a cyano-substituted methoxylane derivative. In the first step, we brominated both methyl groups. Then we performed a Gabriel synthesis with potassium phthalimide. And then we removed the phthalimide protection by treatment with methylamine. And under these basic conditions, the nitrogen is attacking the carbonyl of the nitrile group, and it comes to a spontaneous ring closure. And so we obtained this desired amino isoindole derivative with this amino methyl substitution. We gave them the abbreviation AMIA. It stays for amino methyl isoindole amine. Yes. And the rest of the molecule was synthesized by solid phase peptide synthesis. We started with the loading of a chloro-trityl chloride resin with the FMOC protected canavanine. It's Spock protected in the side chain. And then we were coming to this resin bound intermediate 3. And then we added 3 rounds of standard solid phase peptide synthesis. In the first round, we coupled FMOC tertiary leucine. Second round, again, FMOC canavanine. And as third residue, this FMOC amino methyl phenyl acetic acid. Then we obtained this resin-bound intermediate 4. Afterwards, we removed the FMOC group, treated the free amine with dibocpyrazole carboxamidine. And so we constructed the guanidino group. Then we cleaved the whole peptide from the resin under mild acidic conditions with 1% TFA. This maintains all the protecting groups, and then we coupled this P1 residue and isolated this by preparative HPLC. The inhibitor was synthesized by my PhD student Roman Lange, which is now a postdoc in my group. And the compound with this amaya residue had a Ki value of 7.1 picomolar. It's also very potent. And also in that case, all 4 mice survived an IP dose of 50 milligram per kilogram, but 1 of 4 died at 20 milligram per kilogram. So we cannot say that this compound with this amaya residue is a little bit more safe than the compounds with the benzamidine. And this figure or the photo simply shows you the superimposition of an inhibitor which contains this new amino isoindole group in P1 position, colored here with the carbon atoms in dark green. And this is superimposed with an inhibitor containing the benzamidine, which we used in all previous inhibitors with the carbon atoms in light green. It adopts completely the same conformation when bound to furin. So, and what is the reason that these compounds are less toxic? I already explained that we tried to reduce the number of strongly basic groups. And it's important to know that furin is making this activation of the viral glycoproteins usually when it is located in the trans-Golgi network. And in the trans-Golgi network, we have a pH of around 6. And we always assumed that the toxic properties of our inhibitors are coming from the targeting of an off-target somewhere in the circulation. In the circulation in blood, we have an pH value of around 7.4. And if you simply calculate the protonation state of these inhibitor at pH 7.4, you can simply use the Henderson-Hasselbalch equation, you get as a result that only approximately 10% of the inhibitor should be fully fourfold protonated. And we assume that the fourfold protonated form is responsible for the toxicity. Whereas if you calculate the protonation state at pH 6, there we are around 1 unit below the pKa value of 7 for this cannabinoid sidechain, then approximately 80% of the inhibitor should be fully fourfold protonated. Yes, and this is important that the compound can act as inhibitor, as it needs this fourfold protonation to inhibit furin. Yes, but we assume that the off-target exists in the circulation at a higher pH of 7.4, and there we have only a small amount of this compound in this completely protonated state. And during that time, I heard a talk of a colleague from the neighbor university from Marburg. It's the University of Gießen. It was given by Franco Falcone. And he spoke about a special mast cell related G protein coupled receptor member X2. And he explained that this receptor can be addressed by cationic antimicrobial peptides. Yes? And it was also known from previous studies with thrombin inhibitors that certain benzamidine derivatives induce a rapid histamine release from mast cells. Histamine can bind to the histamine H1 receptor, then it comes to a release of nitrogen monoxide, and this subsequently results in a vasodilatation. The vasodilatation has a rapid drop in arterial blood pressure as consequence, and this can lead to mortality. This was described for benzamidine-derived thrombin inhibitors. And it was also described in a later publication from the same group that this histamine release works through an activation of a GI protein. An inhibitory G protein, this can be inhibited by pertussis toxin, and they could demonstrate that in presence of pertussis toxin, it's not coming to a histamine release. And this mast cell-related receptor, about which my colleague spoke, is also a GI-coupled receptor. And that's why I asked him if he can test if our compounds can also activate this receptor, which finally, after activation, leads to an histamine release, drop in blood pressure, and mortality. And interestingly, he had a nice test system available. He had a rat basophilic leukemia cell line, which was stably transfected with a neuropeptide Y plasmid, which was, also the neuropeptide Y was coupled with a red fluorescence protein as a kind of reporter. And this protein construct is also stored in granules like histamine, and this cell line was also stably transfected with this mast cell related receptor, a G protein coupled receptor member X2. And simply, if you gave some receptor activating compounds to the cell line, then it comes to an activation of the receptor, and the content of the granules, this neuropeptide Y connected with a red fluorescence reporter protein, is released into the supernatant, and you can simply measure the fluorescence of the red fluorescence protein. And Franco Falcone tested some of our compounds, this inhibitor MI1148, which was the most toxic one. And at the left side, you see negative control. You see a very low fluorescence. Then he has used a positive control, this compound with the label 48-80. It's a compound consisting of a methoxyphenylethylamine, which is polymerized by formalin. And this is known as a very strong activator of this mast cell-related G-protein coupled receptor member X2. Yes, and when you add this compound, you get a very significant fluorescence, and also in presence of our more toxic inhibitors, we also obtain a significant release or increase of the fluorescence. So it means that the receptor is activated by this compound. And when we use the compounds with the significantly reduced toxicity, which contain these two cannabinine residues, the toxic compounds contains the arginines here in P2 and P4 position, then we have seen a very weak increase of the fluorescence. So it means, or it seems, that this compound cannot activate efficiently this mast cell-related receptor. Yes? And the same applies for this compound 2421, where we have only an amino-methyl substitution. This has also a very low toxicity, and this can also not activate this receptor. Only this toxic compounds activates this receptor. And therefore, we believe, at least, that this receptor could be the potential off-target of our polybasic furin inhibitors. And if we don't target or address this in receptor, then we have a reduced toxicity problem. And here you simply see some tests of these compounds with several furin-dependent viruses. These experiments were done in the group from Ralf Bartenschlager in Heidelberg. He had a cell culture system infected with dengue viruses, and he used the inhibitor 1148 and the less toxic compound with the cannabinines 1851, and we see a nice concentration dependent decrease of the viral titers. But we have to say we need concentrations around 4 to 8 micromolar for a significant reduction of the virus titers by approximately 3 to 4 log steps. Yeah? And this is necessary to achieve a significant antiviral effect. And of course, these concentrations are approximately 100,000-fold higher than the pure Ki values against furin, most likely due to a poor cell permeability of this very polar compounds. Ribavirin, it's a nucleoside analog, which is a standard antiviral compound, was simply used as reference, and it was even used or required at higher concentrations. Yes? And it's an approved drug. It was previously used at least for the treatment of hepatitis C virus infections, but meanwhile there exists better drugs against hepatitis C virus treatments. Okay, very similar results we obtained with a different flavivirus. This is the West Nile virus. The viral titers are shown on the y-axis. And also in that case, we need concentrations around 8 micromolar to achieve a significant antiviral effect. Then these are experiments with the respiratory syncytial virus. These experiments were done in Marburg in the group of Eva Friebertshäuser. The respiratory syncytial virus can lead to pneumonia and bronchiolitis, so it makes simply an infection, what the name says, of the respiratory tract. And it is especially harmful for very young children. And each year worldwide die approximately 60,000 children in the age below 6 months. Yes, and you have each year approximately 3 million hospitalizations. So it is a severe virus. And in that case, we also used concentrations between 10 and 1 micromolar to achieve an antiviral effect. And in that case, our compound with the cannabinine residues, which was less toxic, was even more potent than the analog with the 2 arginine residues. And I have to say, I forgot to mention it, in all cell culture assays, which we did before the compound have seen the first time in mice, they were completely well tolerable, all these compounds. This compound with the arginines, nothing did not affect the cells. Yes, they were very, very well tolerated. They showed no sign of cytotoxicity, but it's different in an animal. Okay, so we need here with this compound concentrations of between 1 and 2.5 micromolar. So it was relatively efficient. Okay, and this was the first part of my talk, and now I'm coming to this new non-peptidic inhibitors. There was a patent application from GlaxoSmithKline at the end of 2019. I have seen it somewhere at the beginning of 2021. And they claimed completely different structures as furin inhibitors. And so at that time, it was nearly impossible for us to explain their binding mode. Yes? And the first structure we have seen in a preprint of this publication, which appeared finally in 2022. The preprint appeared somewhere in 2021. And of course, we did not like to synthesize the same compounds. We wanted to modify them a little bit. And then we had the idea to replace the sp3 hybridized oxygen on the right inhibitor arm simply by a methylane group. And the synthesis was relatively straightforward. I have shown it here. We started with a bromo-substituted isophthalic acid, performed a Suzuki coupling, and obtained this bis-chloro-substituted biphenyl intermediate 3. Then we reduced both carboxyl groups with a borane THF complex, obtained the bis-hydroxy derivative, which was then later converted with reaction with phosphorus tribromide into the bis-bromide. And this work was done by Roman Lange. And when we treated this intermediate 5 with an excess of 1 amine, we obtained symmetric inhibitors. So one of these first analogs had a Ki value of 77 nanomolar. It was not bad, but also not very exciting. And if we treat this intermediate with 2 different amines, then we always get a mixture of 3 compounds, 2 symmetric compounds, and 1 asymmetric compound. And the asymmetric compound usually was the more potent inhibitor. Also this compound here inhibits furin with a Ki value of 12 nanomolar. And this simply shows you a reaction control by HPLC. And if you treat this bis-bromide with 2 different amines, the different amines differ also in hydrophobicity. And therefore, the consequence is that the asymmetric product, which is shown here in the middle, elutes always as a second peak. The first peak which you obtained is this product, which contains twice the more hydrophilic amine. It's this derivative. And the more hydrophobic peak contains twice this amine, which is protected by this more hydrophobic Bock group. Yes? And either you separate these 3 compounds on a protected stage. This is the signal from the starting material. It's nearly completely gone. Then we have 2 little side products. But in some cases, we also separated the products by preparative HPLC at the end after removal of the protecting groups. And so, of course, this compound here, which doesn't contain any Bock group, it stays at the same, it elutes at the same time, around 26 minutes. And again, here in the middle, we have the asymmetric product. And normally, if it works in a normal way, you should have obtained these 3 peaks in a 1:2:1 ratio. But this is not always the case. It depends on the reactivity of the amines. But usually, we have no problems to separate these 3 peaks. And it's a very straightforward synthesis of these compounds. And then I prepared a little movie to explain the binding mode of these compounds. At the beginning, you see the complex of furin with our peptidic inhibitor with this amide residue in the P1 position. Then I show this symmetric compound, and then comes an additional inhibitor. I hope it works. I tested it at the beginning. As we zoom into the active site, this is the so-called S1 specificity pocket in which the amide residue binds this amino isoindole group. Yes. And this is the active site cleft of furin. And then I will stop. In all these crystal structures of furin in complex with many different inhibitors, which we were known from our previous studies, this tryptophan of furin is always placed in this position. Yes. Also in ligand-free furin, there exist crystal structures which are suited for soaking. Also these crystals, the tryptophan residue is always in this position. And very surprisingly, when the structure was solved in complex with this compounds with the biphenyl moiety, the tryptophan rotates away from its normal standard position. It makes a rotation by nearly 180 degrees. And the S1 pocket is closed now by this side chain of the tryptophan. And a new hydrophobic pocket is formed on the furin surface. You see it here very nicely. It's a beautiful binding pocket for this dichloro substituted biphenyl residue. And it fits perfectly into this pocket. And we see additional interactions of this nitrogen from the left arm to a glutamic acid and the tyrosine side chain. Then an interaction from the nitrogen of the pyridyl group via water molecules to two aspartic acid residues. And now I superimposed the structure with this inhibitor, where we incorporated a B-piperidine moiety as right inhibitor arm, which was acylated simply by a glycine residue. And then we see that the amino group of the glycine makes a direct polar contact to an aspartic acid side chain and to a carbonyl group, and additional polar contacts to some water molecules. These are the interactions on the left inhibitor arm, and below the inhibitor, we have a whole bunch of a water network, which makes many polar contacts. Okay, and all these crystal structures were determined by Sven Dahms. I mentioned before, he was initially a postdoc in Jena in the group of Manuel Thahn, and later when Manuel retired, Sven moved to Salzburg into the group of Hans Brandstetter, and luckily he continues with his work on the crystal structure determination of furin. And this is a view on this hydrophobic pocket. It's surrounded by 3 leucine residues, and we have one methionine and one I think it was an alanine residue and one valine, yes. But these are all hydrophobic side chains, and that's why this bis-chloro substituted biphenyl group binds perfectly into this hydrophobic pocket. Okay, this is a crystal structure of a compound with a B-piperidine group as right inhibitor arm, and you see that the terminal piperidine ring is missing. So it's a clear indication that this part of the inhibitor seems to be very flexible. So Sven could not detect and define electron density for this part of the molecule. But the situation was different when we acylated this B-piperidine with this glycine. I have shown this in this movie. The glycine works like an anchor, yes, and makes these many polar contacts. And then we had seen a very well-defined electron density for the whole right part of the inhibitor. And we tried to modify the left side of the compounds. We always kept the B-piperidine as right inhibitor arm, but you see there are no major differences in the Ki value. They are all very similar, as we made many compounds. And the most potent inhibitor of this first series, where we had here on this as first ring on the right inhibitor arm, a piperidine or piperazine, was this compound 2471 with a Ki value of 1.7 nanomolar for furin. And you see from this pyridyl nitrogen, we have some water-mediated contacts to these furin residues. There are 2 aspartic acids and an asparagine, and here's even a 3rd aspartic acid. And the next PhD student which works in the field of furin inhibitors is Charlotte Boller, and she changed the first ring from a piperidine or piperazine to an aniline-like structure. And this was one of her first compounds, which possesses a very similar potency, also with a Ki value of 0.8 nanomolar. And we also see again several water-mediated polar contacts to an aspartic acid residue from furin and even to an arginine side chain, as in this position 193. And this is this tryptophan residue, which is rotated from its usual standard position to this new position and opens this hydrophobic core, also this hydrophobic hole. And very surprisingly, when we simply replace this carbon atom in this terminal piperidine ring by a nitrogen, now we have a piperazine. As a consequence, this resulted in a shift of this anilinic nitrogen by nearly 2 angstrom, and subsequently also the terminal nitrogen shifted nearly 2 angstrom upwards. And as a consequence, we have now a direct salt bridge from this piperazine, this is the terminal ring, from this nitrogen to this aspartic acid side chain. Yes, it's 2.8 angstrom the distance, and this is probably one of the reasons that this compound has a further improved potency of around 1.1 nanomolar, and we see an additional polar contact from this nitrogen to this glycine carbonyl group glycine 255. And a very poor potency we observe when we replace the anilinic NH group simply by an oxygen. The affinity dropped by a factor of 1000. This is only a micromolar inhibitor. So this aniline is an important structural element in these inhibitors. I have only one slide showing how we are doing the enzyme kinetic analysis for this inhibitor, which I have shown at the end with this Ki value of 1.1 nanomolar. We observed in so-called slow binding behavior. You see here a progress curve. On the x-axis, it's the time. On the y-axis, the fluorescence. We incubated enzyme inhibitor and the fluorogenic substrate. And at the end, we have a linear steady state. From the linear slope, we can calculate using this equation a steady state velocity. And in the initial part, we have a curved, a time dependency, and we can calculate by using this equation a first order rate constant for each inhibitor concentrations. And then if we plot the steady state velocities as a function of the inhibitor concentration, we can simply calculate a Ki value as a parameter using equation 2. And if we plot the first order rate constants K observed from the inhibitor concentration using this equation 3, we can calculate from the slope of this linear relationship the association rate constant K on. This is the reaction rate constant for the reaction of the enzyme with the inhibitor. And if you know the Ki value from this plot, the K on value, the association rate constant from this plot, you can calculate with equation 4 simply the dissociation rate constant K off. Yes. And when we compare these kinetic data, which we obtained for a peptidic inhibitor and for this non-peptidic inhibitor with this structure, we use this peptidic inhibitor, you can compare the Ki values and you see immediately that this non-peptidic inhibitor is approximately a 150-fold less potent furin inhibitor. But the reason for this is that the association rate constant is considerably smaller. It's approximately 330 times smaller compared with the peptidic inhibitors. And we did some measurements also for other inhibitors. We always obtain similar association rate constants for these non-peptidic inhibitors. We simply assume that it depends from the rotation of this tryptophan residue. This is the rate limiting step for the binding. Yes. And surprisingly, the dissociation rate constant K off is even smaller in case of this non-peptidic inhibitor. It means if the complex is formed, then the dissociation is reduced compared with the peptidic inhibitors. And from this dissociation rate constant K off, you can calculate the so-called residence time. It's simply the reciprocal value of this dissociation rate constant. And it says you that the complex with the non-peptidic inhibitor is even more stable than the complex with the peptidic inhibitor. We sent also these compounds to our cooperation partners from the virology. And in that case, it was these compounds were tested with an mouse-adapted influenza strain with the name SC35M. It's an H7N7 strain. Originally, it was isolated, I think, from a seal on the coast of Massachusetts. And we see with all these compounds, we also need approximately 5 micromolar concentrations of the inhibitor to obtain a significant reduction of the viral titers. So we tested 1 micromolar in red. In blue is the curve with, or the growth curve at different times with a 2.5 micromolar inhibitor concentration, and in green with the 5 micromolar inhibitor. The control is in black. And surprisingly, also a little bit, the strongest effect we have seen with this compound, which even has a little bit lower or weaker Ki value than this compound in the middle. And yes, so there was not a perfect correlation of the inhibitory strength against furin and the antiviral efficacy. And then we tested also this compound 3129 with this aniline structure, and the experiments were done by Konstantin Bloch in the group from Eva Friebertshäuser, which is now the group leader in the Institute of Virology. And we see at least at 5 micromolar, there is a nearly complete inhibition of the virus growth. So the data points are directly on the baseline, but they have a level of detection around 100 plaque forming units. Yes, this is the lower level of detection. So it's at this concentration, it's relatively potent. Okay, then I have only the summary at the end. I can say that we could develop over a long period of time highly potent peptidic and peptidomimetic furin inhibitors. And when we replace the arginine residues in P2 and P4 position by cannabinine, we were able to reduce the toxicity of these compounds considerably in mice. And we hope that we meanwhile could identify the mast cell related G protein coupled receptor member X2 as the potential off target of these peptidic furin inhibitor, which causes the toxicity. And we need usually concentrations between 1 and 5 micromolar to achieve a significant antiviral effect against furin-dependent viruses. And then we prepared several new non-peptidic inhibitors. I could not show all structures what we have done. We determined several crystal structures in complex with furin, which are shown here. It's a superimposition of all these structures which we have. And we need also for these non-peptidic compounds similar concentrations in the range of 5 micromolar to achieve a significant antiviral effect. And at the end, I have to thank many people which contributed to these results. First, all the people from my group which were dealing with the development of furin inhibitors. And my thank goes also to my former colleague Gerhard Klebe. He is now also retired. In his group, Sven could perform this fragment screening, and Gerhard helped us all the time. And then my special thank goes also to the group of Eva Friebertshäuser in the Institute of Virology. Originally, I started my collaboration with Wolfgang Garten. He was the former supervisor of Eva. Now Eva is the group leader. And then my special thank goes to Sven Dahms, which is delivering us always these nice crystal structures. So it helps us a lot of understanding the binding mode of these compounds. And he was former in the group from Manuel Tan, which is meanwhile retired. Then he moved to the group of Hans Brandstätter to the Paris-Lautron University in Salzburg. Franco Falcone made these experiments with this mast cell-related receptor for us to identify the potential off-target of furin. The furin which I am using, I obtained in 2008 from Iris Lindberg and from the University of Maryland. It is still active, and for enzyme kinetics, you do not need a lot of compound. Yes, for the crystallization, of course, you need a lot of protein, but Sven can produce it also. And in the former years, we were supported by 2 grants from the state of Hesse. There is a special program which is called LOEWE. And these grants are now finished. And since the beginning of this year, we get some funding from the European community within the project Vigilant. Yes? And the aim of Vigilant is to use combinations of different protease inhibitors, not only of furin inhibitors. We want also address inhibitors of cysteine proteases and use them in combination with the furin inhibitors for antiviral therapy. Yes? Perhaps we hope that this is a more effective way to achieve antiviral effects. And in addition, we also deal in this project with inhibitors of the ER transport. Also there are some translocons where the proteins after protein synthesis were transported into the endoplasmatic reticulum. And if you block this way, then you can, of course, also achieve an antiviral effect. And this is the final slide of my group. This was at the end of last year. And this was in this summer when we made a kayak trip on the River Lahn. And I'm finished now. I'm happy for questions.
Thank you very much for your amazing lecture. And it's a mixture of medicinal chemistry, pharmaceutical chemistry, and of course, synthetical chemistry and biochemistry. So we have a hand now. Victoria, please.
Thank you so much, Torsten. Amazing presentation. It is fantastic job. And my question is, as I think, well, most of your experiments were in isolated enzyme in vitro.
Yes, this is what we can do in my group. We are testing, we are making enzyme kinetic measurements.
And what do you think about delivery of these compounds into Golgi apparatus? Because furin expresses in Golgi apparatus.
Yes, it's correct. There is one paper from Stefan Kunz, I think it was from 2017. One of my co-workers, Cornelia Hardes, visited his group, and there they tested many of our peptidic inhibitors at that time for the ability to reach the furin in the Golgi apparatus.
Mhm.
And in this group, they have developed a sensor. It was a protein which contains a furin cleavage sites, and at the end terminus, it contains a luciferase. And this protein was expressed in the Golgi, and when the inhibitors reach this protein, then they cleave this protein, and the luciferase is released into the cell supernatant. And they could measure the released luciferase simply with an activity measurement. And with this method, they could test the ability of the different inhibitors to reach the furin directly in the trans-Golgi network. And for the most potent compounds at that time, it was 1148 in cell culture. Yes, at least at this time, we were already known that it is toxic in mice. This showed also the most effective inhibition of this sensor protein in the Golgi. And it's not, as we cannot exactly say how much of the inhibitor can reach the furin. But what we can say, if you have such a high potency with a Ki value in the picomolar range, then you can assume that everything of the inhibitor which comes into the Golgi, to the furin, the neighborhood of furin, then it leads also to an inhibition. You can see it also. I have even an enzyme kinetic slide. These peptidic inhibitors were analyzed under so-called tight binding conditions. So you see it here, if we have a 1 nanomolar concentration of the inhibitor, the enzyme is nearly completely blocked. Yes,
Yes.
even if we below nanomolar, yes, because it's so potent. And in that case, these data were fitted to this equation. It's the so-called Morrison equation. It depends from the enzyme concentration. The drawback of this is you have to know the enzyme concentration to fit the data. But otherwise, if this is completely wrong, then it's impossible to fit the data. So we tested this as good as possible, and you get here an apparent Ki value, then you have corrected by the substrate protecting term. Because if you need a higher substrate concentration, then you get a weaker inhibition. But at least we assume that everything which can reach the furin in the trans-Golgi, it is probably a very small amount of the inhibitor. And that's why this is the reason why we need approximately 100,000 times higher concentrations in cell culture, yes, to achieve an antiviral effect. But at least something seems to reach the furin. Yes, this I can say based on this work with this furin-sensitive sensor protein. There is a paper with Stefan Kunz in 2017 or 18 in Fab's Letters, which describes this.
And thank you. Yeah, I understand you. And my second question is, it is known that activity of expression of furin depends of some other factors, such as a concentration of cholesterol. What do you think about influence of cholesterol on activity of furin in presence of your substances?
Okay, so I cannot say a lot to or nothing to this. I remember only that we tried once. It was a different project. It was when we were dealing with inhibitors of the West Nile virus protease. We covalently coupled cholesterol to our inhibitors. Simply, we wanted to improve the cell permeability. Yes, because in the lipid envelope, you have a lot of cholesterol. And the consequence was that we always obtained a significant inhibition of the protease, because the protease of these flaviviruses is sitting in invaginations of the endoplasmatic reticulum, but in the cytosolic side of the cell. Yes, and therefore the compound has to pass the cell membrane, and then it's in the cytosol and it can reach the protease. But what we found with this cholesterol-bound inhibitors, always a very strong inhibition until a concentration around 1 micromolar, and then below 1 micromolar, we had a very steep increase in the enzyme activity, so no inhibition anymore. And we believe that it was an unspecific attachment of cholesterol to the protein, because if you measure IC50 curves, normally with normal competitive inhibitor, you get a slope factor of 1. And with this cholesterol, the slope factor was 4. And then we simply modified also the inhibitor part. We simply coupled a simple arginine residue, which cannot inhibit the protease. We obtained absolutely the same inhibitory behavior, a very strong inhibition until a concentration of 5 micromolar or something. I don't know, remember exactly. So it means it was a completely unspecific effect with the cholesterol. But we never added cholesterol to our assays, because it's a very hydrophobic compound. I don't know if it forms micelles and something. It would probably disturb the enzyme kinetic measurements. And I don't see the reason why we should add cholesterol for experiments with furin.
Yes.
And we are happy if everything is well soluble. Yes, and cholesterol, it's a very hydrophobic compound. And we did these experiments when we modified our inhibitors. We also attached all of these fatty acids. It is described in this JPC paper from 2012. We simply added an acetyl residue, butyric acid. We went up to 20 carbon atoms. The problem was then always there was some kind of micelle formation. Yes, and this had an influence on the inhibitory potency. It's described in this 2012 paper.
Thank you. Thank you.
Thank you very much. Mykola, please.
Hello again, Torsten. Thank you very much for your very exciting lecture, exciting story of your adventures with all these very interesting molecules. And first question is about this hydrophobic pocket that dichlorophenilyl residue occupies in the target. Is it the same pocket that is occupied by tert-butyl group or different one?
No,
the peptide?
The tertia-butyl group in the peptide has no binding pocket. I had shown it at the beginning.
Yes, now I remember it. It was like sticking out of there.
It was sticking into the solvent. Yes, it's there is no binding pocket. The advantage of this tertiary butyl group is simply that it makes a fixation of the conformation of the beta sheet backbone. of the peptidic structures. And there is a very nice paper from the Fairley group from Australia, I think from 2002 or 2003. The title is Proteases Recognize Their Substrates in Form of antiparallel beta sheet or based on antiparallel beta sheet interaction. So it means if you stabilize this conformation but by a bulky side chain in case of tertiary leucine, yes, you stabilize the inhibitor conformation. And if it's the correct conformation which you stabilize, then you get an improved potency. Yes, and
okay.
This is not, it's very interesting that you find these tertiary leucine, yes, in several marketed protease inhibitors. Yes, the first three are inhibitors of the hepatitis C virus protease. Atazanavir is from the HIV protease, and nirmatrelvir, I have here shown the structure. It's from Pfizer. It's also in P3 position of the coronavirus main protease. In P1 position, they have here this cyclic lactam-like glutamine derivative with a nitrile group, because this coronavirus protease is a cysteine protease. And the cysteine, in contrast to the serine protease, the cysteine exists as a thiolate, already preformed. It can attack the nitrile. It forms such an imidothioester, a covalent bond, which contributes to the potency. And in P2 position here, we have a proline analog and this tertiary leucine as P3 residue also. And if you look at the crystal structure, you have also seen antiparallel beta bond, beta sheet in interaction with the protease.
Thank you. And the next question, that you two like mainstream directions of research are going without crossing, and I was waiting for kind of taking some structural information from, for instance, peptide type inhibitors and using it in this new derivatives. Do you think some guide in this direction?
I did not exactly understand your question. Can you repeat it, please? It was broken, your voice.
Why don't you use some structural information from the peptide type of inhibitors that you developed for development new type of not peptide inhibitors? So this two directions of your research going in parallel, they do not interact. Didn't you try to think in this direction?
Yes, I was thinking in this direction, and probably you can see it from one slide. Wait a moment. I have to find it. It's this. I have to stop this, and I have to. Wait a moment. This slide. In this case, you see that this nitrogen from this aniline-like structure interacts with the carbonyl group of glycine 255. And from our peptidic inhibitors, we know that this carbonyl group is addressed by the NH of the tertiary leucine from the peptidic inhibitors. And that's why I asked one of my PhD students to incorporate here tertiary leucine instead of a ring structure. Yes? And I simply hoped that then with the carbonyl group of the tertiary leucine, we can address this NH group of this lysine. Very similar like in these peptidic inhibitors, but the result was a relatively weakly potent compound. I think we had a Ki value around 0.1 or 0.3 micromolar. Yes, also it was significantly less potent than this, but this interaction to the carbonyl group of glycine 255 is normally done in this peptidic inhibitors by the NH group of this P3 residue. But it was at least this single derivative which we have synthesized was not very potent. Yes, but of course we think also in this direction, what you can learn from these peptide structures to use it here. Yes, to use it for the non-peptidic inhibitors to improve them. But there are some other possibilities, but I cannot show everything here in the moment. Yes.
And maybe the last question is possible, if possible. You said also there are problems with cell permeability for peptide type of inhibitors, and this is clearly touched also. That was already discussed with Victoria questions. But I want to ask in different way. Did you try to apply in vivo models for analysis of most potent?
No. So far, you mean something like Pampa assay or something? Yes. There are some permeability assays or this CACO assay or such things. Do you think about this?
I'm thinking about like making a model that infected with viruses and to try to treat it directly with inhibitors.
But what I have shown you here, these cell culture assays, there were the cell cultures infected by viruses. Yes, and then the virus titers were determined. So it is like a model how effective this compound is in a cell culture system. Yeah, it's not in vivo. Cell culture is not in vivo. Yes, and a problem is also at one side you believe that these many basic groups are bad for cell permeability. Yes, but otherwise, if you think many of this cell permeabilization reagents, these are very often polyimines. These are also polybasic compounds which you can use to transfect cells, for instance, with nucleic acids and something. Yes, but a common problem with multi-basic compounds is that they are coming into the cell, into endosomes, and there they stick in the endosome and they are not released. So our compounds must to come to the furin in the Golgi. So if you have the compounds in an endosome, there are several ways where endosomes can travel. It can come to a further maturation of the endosomes. They are then lysosomes and there everything is degraded. They can be transported back to the cell surface. Yeah, then it's also not this what we want, but also endosomes can probably migrate to other enveloped organelles, also to the trans-Golgi network. And so probably the compounds are coming to the trans-Golgi network. Yes, because membranous particles, they can travel within the cell.
Thank you.
I can also say we prepared once where we incorporated a lysine residue in P3 position, and to this lysine we attached a fluorescein marker. Yes, and we looked for the fluorescence in the cell, and we have seen a little bit fluorescence around the nucleus. Yes, also I have somewhere a slide, but I have not it here in my presentation. But it was not a continuous, we could not see a continuous fluorescence around all cells. Only for some certain cells there was fluorescence seen around the nucleus, yes, where the trans-Golgi is located. But if you add a fluorescence label, for instance, fluorescein, yes, it's a very big label. It completely modifies your compound, yes. Also, I don't know exactly the molecular weight, but it's around, I assume 350 or 400 of fluorescein. It's a big label.
Thank you very much. Very interesting.
Thank you. Alexander, please, I think it's the last question.
Okay, thank you, Michael. Thank you very much for very good healing. So I'm also impressed by your first part of presentation about peptidomimetic inhibitors. And when you discuss the role of this central bulky carbutyl group, you presented analog of nirmatrelvir from Pfizer. But this inhibitor is actually known to be covalent inhibitor. So my question is it possible to develop covalent inhibitor of furin? Is there any amino acid which can be covalently blocked by inhibitor?
Of course, this one crystal structure, which I had shown at the beginning, this was a chloromethylketone, and this is contains an warhead which makes an irreversible bond. But it's not perfectly shown here. No, I modified this. Even the reference is wrong. As the first crystal structure, this is a compound from us because here I see the benzamidine. I have made a mistake. The first crystal structure published in 2003. It was determined in the group in Martinsried, in the group from Wolfram Bode. It was also Manuel Tan was involved. It was a complex with an irreversible inhibitor. But chloromethylketones are not suited for drugs. They have tested them in the field of thrombin inhibitors. They are in vivo. They have a half-life from approximately three minutes or even less, because chloromethylketone easily reacts with sulfur-containing compounds like glutathione and so. But a nitrile is also a possible group for certain serine proteases. Indeed, it was published originally also in the 1980s for thrombin inhibitors. There they prepared a nitrile derivative of arginine, and yeah, it was fairly potent. But the serine is not so nucleophilic like the thiolate of an cysteine protease. In the cysteine protease, there is also beside the cysteine, there is also a histidine residue. And there is a preformed thiolate imidazolium formed. And the thiolate is much more nucleophilic, attacks much more easily the nitrile group than a serine is doing this. But you find nitriles also in inhibitors of serine proteases, for instance, in the so-called gliptins. These are inhibitors of the dipeptidyl peptidase 4 for the treatment of diabetes. And this was the topic of my PhD work. And we also, I synthesized inhibitors of this DPP4. And I even wrote in my PhD work, at the end, we had always to write an outlook. And I suggested, because I was known this paper with this nitrile-containing thrombin inhibitors, that you should try to make a nitrile-derived inhibitor of DPP4. But of course, I left the group where I did my PhD work, and 3 years later, it was described by other people. So I had written that it would be useful to do it, but I never did it, and that's the problem. So, and now there are several inhibitors, also approved drugs for dipeptidyl peptidase 4. These are called gliptins. I don't know if you know them. And I think we tried it once to make an arginine nitrile, but it was not good. Yes. And because the furin also the serine is not so strong nucleophilic like an cysteine group. But of course, there are many other warheads known which make covalent bonds for serine proteases. But for instance, boronic acid derivatives. Yeah, but the synthesis of a boronic acid of arginine is not such a simple task. It's published, it's possible, but we have never tried it because it's a very troublesome synthesis. You can use also other ketones. Also, and even, yes, I remember I did it in my postdoc time. I synthesized or I modified chloromethyl ketones simply by reactions with amino acids. So I obtained amino methyl ketones. And these are very potent inhibitors. But very often, if you have such warheads which make covalent bonds, you lose very often a little bit specificity for a certain protease. Yes, because it's a structural feature which then helps to attack many proteases of the same family. Yes.
Yeah,
and in case of this nirmatrelvir, the affinity is achieved simply or at a very strong manner by this cyclic glutamine derivative. Yeah, because if you simply look on the side chain, it looks like glutamine, but the terminal amide is involved in such a lactam ring. And this was known that it's very well a short time after the SARS-CoV-1 outbreak in 2002. Then the groups developed this group, and what Pfizer did, they combined simply this residue with this part, and the P2 and P3 residue. It's exactly the same like in boceprevir. Yes. Also what they simply did at that time, they combined this known P1 residue, which was known from the in the SARS-CoV-1, it was not a pandemic, it was an epidemic in Asia. Yes, and then they combined it with this P2 and P3 residue known from boceprevir. And in the Chinese analog, in the simnotrelvir, they used a different proline derivative, which is known from one of these inhibitors of the angiotensin converting enzyme. There is such a sulfur connection here. And of course, they use a trifluoacetyl group. We are using often a phenylacetyl group. We use a phenylacetyl group very often because we see then derivatives very easily on the HPLC. If we have, for instance, coupled amino acids which have no aromatic system. Yes, there we have sometimes problems to detect them on the analytical HPLC, and therefore it's always good if you have an aromatic system in your compounds for HPLC analysis.
Okay, thank
but
you.
I'm pretty sure there are possibilities to make covalent inhibitors, but their synthesis would be more difficult.
Okay, thank you.
Thank you very much. Thank you again for your very amazing and interesting lecture. Now I have some feedbacks from our participants, and they said that this was really interesting and useful for our community. And dear colleagues, also for you, thank you for your participation in our seminar today.
Okay, thank you very much.
Thank you. Have a nice evening.
You too. Okay, bye.
Goodbye.